All right, so now let's talk about the link between linked genes and crossing over. So we know that linked genes are those that are on the same chromosome, and so I would say that genes A and B are linked; they're on the same chromosome, and they're more likely to be inherited together, especially if they're close to each other on that same chromosome. So what crossing over does with linked genes is mixes them up. So here I have my chromatids from one of my parents. So maybe this is the chromosome, the alleles that I got from mom, and these are the ones that I got from dad. And so these are the alleles from mom, the alleles from dad. Same genes, they're both F's and E's and D's. It's just that the alleles, the versions of those genes are a little bit different. So during crossing over, these chiasma are going to form during a process called synapses. And they just kind of stick to each other. There's a lot more detail than that, but we're going to go with they stick to each other for now. And they trade those chunks. So this used to be one of the arms of dad chromosome, one of the arms of mom chromosome. They just trade those two chunks. They get broken off and recombined. And so I would call this recombinant DNA over here, this arm of this chromatid especially, and this one as well. So this one is a little bit of leftover of mom on the rest of this chromosome that's mostly mom. And then I've got this little chunk of dad, and over here vice versa. Now, in reality, these chromosomes are not just going to switch one and one, but in fact, they would be like trade, 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 trade. So there'd be lots of like mom, dad, mom, dad, mom, dad, dad. So it's not just like one little piece that breaks off from the bottom of the chromosome. It's just easiest for us to draw the pictures that way. So recombinant DNA we can notate like this. So this is our idea, our notation for showing linked genes. This individual is heterozygous, big A, big B, little b. And so I could have written that genotype just like that. It's our normal way of writing it. So this doesn't tell me, though, if A's and B's are on the same gene or on the same chromosome. If they're on the same chromosome, if they're linked or not. This, however, does make it pretty clear that A's and B's are linked. And so I like to draw, we talked about this in class, I like to draw chromosomes around those lines so that I'm thinking clearly that this is a chromosome with dominant A and dominant B. This is a different chromosome that I got from my other parent with recessive A and recessive B. And I'm looking for recombinants. I want to see what would happen if they crossed over. And so if that happened, they're going to chunk off here. And so I'll end up with little a, big B on one chromosome. And then my other recombinant would be big A, little b. Now, of course, not all of the chromosomes and all of the cells are going to do this. And so it'll happen just sometimes, which is actually really good for the population because, again, that means that we have an increase in genetic variation. Not every individual is the same. So here's just showing some data about our recombinant DNA. So I've got purple sweet pea plants with long pollen grains crossed with red plants that had short pollen grains. In our F1 generation, that first filial generation, all the plants were purple and long pollened, which leads us to believe that purple and long is definitely dominant. And then in my F2 generation, again, where I take these guys, my heterozygous purple long, and I cross with my heterozygous purple and long, I end up with 70% purple and long, and I have 18% red, and that should be short. I don't know where round came from. So these guys are matches for my parents, that original parent generation. And it makes sense because I have mostly dominant and a little bit recessive because I had these recessive guys in here, allowing that trait to be expressed. But the weird thing that I have in here, very small percent, only 6% are recombinants. So these are my two recombinant phenotypes. Parents were purple and long, or red and short. I didn't have any parents that were purple and short. I didn't have any parents that were red and long. And so these individuals ended up with eggs or sperm that had recombinant DNA. Crossing over occurred between those two genes in the eggs and sperm that resulted in these individuals. So these are our recombinant offspring. These guys are not. These guys are not. Good. Perfect. So we're going to talk about variation really quickly, and then we're going to end this piece of the video. So phenotypic variation falls into two different kinds of categories. It can be discrete or it can be continuous. So discrete simply means that it's categorized. It's this or that. It isn't somewhere in between. Blood type is a great example of discrete variation. We have varieties of blood type. You can be A or B or AB or O, but you can't be like a little A, a little B, whereas you could totally be like kind of tall. You can't be kind of type AB. So that's discrete versus continuous. Height is definitely continuous, where we have really short people and really long people and everything in between. Type AB, there's nothing in between. You're either this or that. Good. Another example of continuous variation is eye color. And this and a lot of our other continuous variation traits are polygenic, which simply means that there are many genes that code for that particular phenotype. We think there's up to 16 different genes that influence human eye color. I'm going from very pale eyes to super rich and dark eyes. And it's related not to actually different colors of pigment. It's all melanin. The same pigment in all of our eyes. It's just a matter of how dense that pigment is loaded into the iris. So the more dense that melanin is, the more dense the cells are, the darker the eye color is going to be. Whereas if things are spread out a lot, which allows for more light scattering, you're going to end up with lighter colored eyes. 16 different genes. And so we don't end up with blue eyes and brown eyes. We have really pale to really rich and everything in between. Continuous variation is also going to be affected by environmental influences. The most obvious one is diet. So your height is going to depend on your diet as well as your genes. So you maybe had the potential to be 5'6", but if you grew up with a very poor diet, if your family didn't have a lot to feed you, you might not be able to reach that potential. So the environment, things like diet, certainly is going to influence how those genes are expressed. Other things that could influence gene expression, internal environment, our diet, our access to vitamins and minerals, but also how hormonal we are. So having a large excretion of hormones or a very small excretion of hormones could influence those traits. Other things, temperature can influence how genes are expressed. Light can influence how genes are expressed. Drugs can influence how genes are expressed. So the environment, as well as what all those alleles that you're born with, can influence those traits, especially those that show continuous variation. And on that, we're going to pause.